This is Nervous System PowerPoint Part 5, the interactions with the muscular system. So this is kind of a new overarching topic. You'll see that at the beginning of a lot of these PowerPoints, they kind of talk about a big picture idea that we need to understand. Those are directly from the standards on the AP test, so it's good to kind of keep them in mind and recognize that these are usually not specific to one unit. They encompass multiple topics on the AP test. And so its interactions and coordination between systems provide essential biological activities, and this example is the interaction between nervous and muscular systems. The contraction of a typical muscle is usually generated in response to something in the nervous system. This really demonstrates the interdependence between the two systems. We need to know a little bit of background about the muscular system. If you have not taken anatomy before, you may not know much about this. So this is going to go a little bit into detail about the muscle system. Every body part or body system is organized into an organ, tissue, cell. We've got our full muscle, which is technically considered an organ. So like my bicep muscle is the organ. The bundle of muscle fibers is going to be the tissue, and then the cell is going to be a single muscle fiber, which is actually a really long multinucleated cell. So it's got multiple nuclei, just a long cell. We then break it down a little bit further. It looks like this. So we go from our cell to a myofibril, which are the tiny, tiny strands within a single muscle cell. In a myofibril, they are divided into segments. A single segment is made up of a light band, a dark band, and a light band. So we'll see that there's two light band end caps and a dark band in the middle. We call the light bands when one section starts and the next, or one ends and the next begins, the Z line. The light, dark, light with the two Z line ends in full is called a sarcomere, and that is the contractile unit of the muscles. The other way we say light, dark, light bands is I bands. That sarcomere is made up of more smaller filaments. So we went from a myofibril to a sarcomere unit to even smaller fibers. They are called thick and thin filaments, also known as myosin and actin filaments, and basically they overlap similar to a zipper. And what will happen is when a muscle contracts, those Z lines, so the end of one sarcomere, are pulled closer together because the actin and the myosin filaments basically squish each other closer, and we'll talk a little bit more about how that happens. Take a minute, do this if you'd like to in your notes. This is the answer key. So something to think about is to take a look at these two images and think about what is the noticeable difference. And obviously the first difference we should see is the length of the sarcomere, and the second is the kind of the density or the darkness of the picture. When we see a muscle contracting, the Z bands get closer together and the actin and the myosin are going to overlap more, making it a denser, more tense cell, which is why when we contract our muscles, it feels thicker, it feels more tense. This is the up-close relaxed version. The muscle is starting to contract, so the Z bands are getting closer, and then totally contracted, the two white Z bands are totally close, and the actin and myosin are totally overlapped. So let's look at this at a molecular level. There is a video on YouTube, look it up if you want to, but this is what's important. So we've got a single strand of myosin, the thick filament, and actually we see in the big picture up top that there are two actin filaments. They are the orange colored ones. So what we see is that in the contraction of the muscle, excuse me, they are unattached when they are relaxed. What happens is the first step is getting ATP into the mix. Basically what happens is ATP is going to arrive. It's a chemical energy source, and we know we need this for muscle contractions. As we go to part 2, we see that the ATP has bound to the myosin head, and when it's just ATP hanging out, it actually prevents the myosin head from touching that orange actin and doing any contraction. We know that ATP is actually adenosine triphosphate. So there are 3 phosphate groups connected to that ATP. And if I were to snap a phosphate group off, that's the release of energy. And so what I see is that when I remove a phosphate, it allows the myosin head to bind to the actin. The ADP and the phosphate leave the head, it binds to the actin, and then it actually pulls it, or it contracts, kind of moves away from itself. The myosin moves away, or the head moves up. And that, in turn, pulls the myosin, which is where we get the overlapping contraction. This is a little bit of a zoomed-in version. Currently, when ATP is bound to the myosin head, it cannot attach to the actin. It is considered a high-energy configuration. Right now, it's not doing anything. ATP is just bound to the head. When it gets dephosphorylized, when I remove the phosphate, it's going to make a cross bridge, which is the connection of the myosin and the actin. And then the myosin head is going to bend upwards or away from the myosin strand, pulling the actin towards the center of the sarcomere, making the whole sarcomere shorter. Then more ATP comes in and it binds to the head again. This is a really cool image of what that looks like. Obviously, it is quite a bit faster than we would imagine, but think about how quickly you just pull your arm up or down to contract the bicep. All of this is happening in a very short amount of time. The other thing to consider is that calcium plays a very big role in the contraction. Essentially what happens is that there is a troponin complex. It is a protein that sits on the actin. Without calcium, muscles cannot contract. When calcium rushes into the cell, it binds to the troponin complex. The myosin binding sites are opened on the actin, and that allows for the contraction. So this is kind of a second or a dual action that's happening at the same time. And if we think back to the nervous system, we know that the arrival of the action potential causes the release of calcium, right? That causes the release of the neurotransmitter. And therefore, that calcium is also used to open up the actin sites for the myosin head to bind to. And so we see this dual relationship between the nervous system and the muscle system. And we know that without that nerve impulse coming to the motor cell or the muscle cell, we won't have a contraction. This is kind of putting the two together. We see that we have the spinal cord in the top left, and we have our motor cells leaving the spinal cord as a nerve and attaching themselves to muscle fibers. The calcium that is released at the very end of the axon terminals of those motor cells, or those motor neurons, I should say, causes the initiation of the contraction in the muscle cells.